Yeah. So first things first, these are not my notes. These notes belong to Dr. Hermias Terefe. Yeah. Moving on. So when we're talking about the macroanatomy of the CNS consists of two parts. Which parts are these? We're saying the CNS consists of two parts. Which are the two parts of the CNS or two sides of the CNS?
We have the brain and the spinal cord.
Yeah, nice. Yeah, the brain and the spinal cord. Yeah. So we'll start with the brain, and we look at the brief anatomy of the brain, then we'll go to physiology. Yeah. So just so the brain, we know the brain is, I think we've looked at the brain before when we were discussing the past papers. So this is more of just an overview again. So starting with where the spinal cord and the brain get in touch, here is where we have our brainstem, starting with the medulla, the pons, then the midbrain there. From the midbrain there, we get our hypothalamus here. Let's see hypothalamus. And of course, hypothalamus being here, you follow it down, you get your pituitary gland. So hypothalamus, hypo means below or inferior to. So above the hypothalamus, we have the thalamus. Yeah. Then from the thalamus, there now we have information that will now go to the cerebrum. Then the brain is surrounded by the brain will be surrounded by the meninges, which protect the brain from toxins, from trauma. We know the arachnoid fluid will flow through this space. This is where we will form our CSF. Yeah, we will mention that choroid plexus and such things. Then the other structure, let me not call it an organ, but a structure we've not mentioned is the corpus callosum. We know the corpus callosum functions to connect the two halves of the brain. We know the brain is divided into two, or rather two hemispheres, right? These two hemispheres here, and the corpus callosum will now connect the left hemisphere and the right hemisphere. Just a second.
Yes.
Yeah, so the corpus callosum, we've said it will connect the two hemispheres, the left and the right hemisphere. Yeah. And let's go back to this image. The skull, we know it will consist of the bony structures that also functions to protect from trauma and also house the brain, right? Of thought. And from the cerebrum, now to take you back to what we had discussed on the other day when we were having the discussion, what are the lobes in the cerebrum and what functions do they play? The lobes in the cerebrum, where are they located and what functions are they associated with? The different lobes and their functions. I won't move until people
There are
talk.
5 lobes. We say that there are 5 lobes: the frontal, parietal, temporal, occipital. 4 actually.
And functions.
The frontal lobe directs movements, also speech association through the cortex, and then the parietal lobe, we said it's mainly receives its area for somatosensory inputs from the skin to the muscles, receptor of the muscles in the body and the head. And then there's also tactical stimulation in the parietal. And then there's body image, speech, expression of thoughts, feelings. And then I'll go to the temporal lobe. There's the auditory area and the olfactory area. The auditory is for hearing, the olfactory is for learning, memory, and emotion. And then the occipital lobe, it receives and interprets the visual inputs. Basically, it does the visual functions and integrity of autonomic informations.
Yeah. Yeah, nice. Yeah, so she's covered the lobes and their functions. Yeah, prefrontal, the parietal, the temporal, and the occipital. Yeah. And now let's go to the division of the nervous system as a whole. So the nervous system will be divided into two: the central nervous system and the peripheral nervous system. The central nervous system, we already said it will compose of the brain and spinal cord only. So all the other nerves that are outside the brain and the spinal cord area will be in the peripheral nervous system. So in the peripheral nervous system, we'll divide it into 2 categories. That's the sensory division and the motor division. The sensory division is which picks up the sensory stimuli, right? Things like touch, hearing, seeing. That's the sensory division. It picks up that information, and then we'll convey that information all the way to the CNS. As Dr. Shafia said, that this information will go all the way to the parietal lobe, where it will be interpreted, and should there be any signal or something that warrants the motor division being activated, the motor division will be activated accordingly. For example, let's say your thermoreceptors have picked up that you are being burned, that the temperature is getting too high, right? All this information will go to the parietal lobe, and then that will be the ascending signal. Then the descending signal will now go through the motor division to your somatic nervous system, so that you can contract your hand to remove your hand, for example, from that burning bush or something, right? Yeah, so the motor division here, majorly we are talking about muscles it's about you. Motor divisions, majorly just muscles. Think of it as muscles, the contracting, the relaxation, the movement, anything that muscles do will fall under this area. And we now know for motor division or for muscles, we have the skeletal muscles, the smooth muscles, and the cardiac muscles, right? For the skeletal muscles, we know the skeletal muscles are the ones under our control. They are the ones under the somatic nervous system, right? The muscles we use for walking, the muscles we use for holding things, you know, the skeletal muscles, the ones under our control, right? If you decide not to walk, you won't walk. And then the autonomic nervous system, here we have the smooth muscles and the cardiac muscles. The smooth muscles and cardiac muscles. Auto means self-driven. So it doesn't need our stimulus it doesn't need our brain to function, right? Things like the heart. We already mentioned the heart. Yeah, the heart will contract and relax without you wanting to contract or relax the heart. The GIT is also made up of smooth muscles. The GIT will move stuff from your esophagus all the way to your rectum without you controlling the movement of that stuff. So that is autonomic, self-driven. And then now under autonomic nervous system, of course, we know now we have the sympathetic division, which will be controlled by norepinephrine, and the parasympathetic division, which is controlled majorly by acetylcholine. So sympathetic, norepinephrine, we know the actions of norepinephrine when it comes to it plays various roles in various tissues. For example, in the eyes, it will function to dilate your pupils, right? In the muscles, it will function to contract your heart more. Parasympathetic, acetylcholine, we don't need to mention much on acetylcholine because we'll focus more on acetylcholine as the discussion moves on. Yeah. So the division of the nervous system, the CNS and the PNS. The CNS, brain, spinal cord, PNS, we have the sensory and motor divisions. We have the autonomic and somatic nervous system. Under the autonomic nervous system, we have the two divisions, the sympathetic division and the parasympathetic divisions. We know the sympathetic is a fight or flight, parasympathetic, rest and digest. Yeah. So this is just another depiction, as we've said, central, brain, spinal cord, peripheral. We have the somatic and autonomic nervous system. Autonomic, it's the involuntary muscles, the cardiac, the smooth. Somatic is the voluntary muscles and will convey the sensory information to the central nervous system. We've already talked about the corpus callosum, is the one that will connect the two hemispheres. Then the lobes, Shafi has already mentioned the frontal, the parietal, the occipital, and the temporal lobe. She's mentioned the functions of the various lobes. So we're not going to repeat those functions. Then moving on to the functions of the various parts of the brain. Now, we've looked at the lobes, but now under the lobes, we have more specific areas, more specific regions. The first one being the cerebral cortex. We know the cerebral cortex, simply put, this is a seat of thought. Yeah, right. This is where you can say this is where the person is housed. You, the person, you are housed at the cerebral cortex. This is where you think. This is where you reason. Yeah, so the cerebral cortex, that's the seat of thought, right? And a lot of things, of course, will be happening in the cerebral cortex, but we can't go into each of all those things. I'll just be trying to give it will consist, the frontal lobe will be part of the cerebral cortex. And in the frontal lobe, we already said, now in the frontal lobe, this is where also or rather, this is where your thinking is also there in the, so in the cerebral cortex of the frontal lobe, yeah, this is where your thinking is. Also, something found in the cerebral cortex is this the Broca's area, which is what directs your speech, and is also involved in thinking about speaking. In short, it is involved with speech, right? The Broca's area, it's involved in speech. When you go to the parietal lobe, already mentioned, it's the somatosensory cortex will be found here, right? The sensory information will be coming here. For the temporal lobe, hearing now it's under the temporal lobe. And also the other thing, also smelling, olfactory, right? The nose. So stimulus related to smell will also be here. Smell and taste. Remember, olfactory deals with both smell and taste. Occipital lobe is you see with your occipital lobe. So when you're hit in the back of your head, you see stars, right? That's a funny way of remembering it. Yeah. So this diagram, and I'm seeing also, okay, I sent a diagram on the group chat, which also summarizes the lobes and their different functions. Yeah, so you can have a look at that. Now, moving on, how is when we are mentioning the brain, we always mentioning the cerebrospinal fluid. So how is the cerebrospinal fluid formed? Anyone? When we're mentioning the brain, we're always mentioning the CSF. When we were mentioning the infections of the brain, we say you do a lumbar puncture, CSF tap. How is the CSF formed?
I don't know how it's formed, but I know it has 4 chambers that are connected to ventricles, and then the ventricles are filled with the fluid. The CSF, it's inside the ventricles. The ventricles.
The ventricle, the 4 ventricles which are interconnected, they will provide that canal whereby the CSF will flow. Yeah, that's true. Anyone who can help out with how is it formed? Where is the origin? Is it from blood? Is blood done something so that it becomes CSF?
Yeah, I think the blood is filtered in the choroid plexus to form now the CSF.
Yeah, nice. Yeah, the blood will be filtered in the choroid plexus, whereby now it will remove, the choroid plexus will be selective, will remove some stuff that shouldn't get in the brain. Yeah, and it will form the CSF. Nice, thank you. So the choroid plexus is at the central, of the formation of the cerebrospinal fluid. Yeah, let me just display this diagram, which help. Okay, I'll share the diagram. Yeah, so the choroid plexus the CSF, it simply comes from blood. Remember, there is no special fluid entering your brain, right? It's the blood that will go, and then the choroid plexus will filter that blood accordingly and get your cerebrospinal fluid. Yeah, so the choroid plexus is critically involved in the formation of the cerebrospinal fluid, which will flow now through the ventricles 1, 2, 3, and 4. At ventricle 4, you can see where ventricle 4 is located, up or just you can see just at the end of the brain there. So there, the ventricle number 4, at the end of ventricle number 4 is whereby kutakuwa more like a U-turn. Just after ventricle number 4, it will just go ahead into the spinal cord for some length, but then take a U-turn and the CSF goes back to the brain. Yeah. So there's the fourth ventricle. Now, looking at the deep line structures, we've looked at the surface structures more. If you just removed someone's skulls, we've looked at what you will find there, the various lobes and the cortex. Now, looking at the deep line structures of the brain, we have the basal ganglia. Now, someone to say something about the basal ganglia. There's a disease that we'll be mentioning the basal ganglia a lot. So, basal ganglia. What do you know about the basal ganglia?
I think that's where acetylcholine neurons are mostly located.
Acetylcholine neurons. Something else about the basal ganglia?
It has some nucleus, and then one of them is substantial nigra. I think it's the one that is affected during is it
During?
I'm trying to remember. Parkinson's disease. Yes.
Yeah, exactly. Parkinson's disease. Yeah, this is where we have the substantia nigra. Yeah. Parkinson's disease, we'll see this being affected a lot. Yeah. And Parkinson's disease, there's a deficiency of which neurotransmitter?
Dopamine.
Yeah, dopamine. So what's the treatment of Parkinson's disease? What do we give? First line treatment?
Levodopa.
Yeah, levodopa. Yeah, nice. So
and also the acetylcholine, it's not basal, it's usually basal, the basal forebrain. Sorry.
I was confirming that first. Thank you for confirming. Yeah, so the basal ganglia, majorly, what you shouldn't remember, what you shouldn't forget about the basal ganglia is that the substantia nigra is here. And you know, substantia nigra, wherever you see it, you start thinking about dopamine. Yeah. Then we go now to the hippocampus and amygdala. Hippocampus, we always say hippocampus has to do with memory. Simply put, hippocampus, memory. That is where your memories are housed, right? Then the amygdala has to do with emotions. The amygdala has to do with emotions. You can see where they are located, the hippocampus and the amygdala. The hippocampus there and the amygdala. So hippocampus, memory, amygdala, there majorly what we have is the emotions. Emotions will be housed in the amygdala. That's why there's this concept known as amygdala hijacking, whereby the amygdala hijacks your brain due to emotions. And it does this by for example, when you see a lion, you're supposed to run, but you freeze. Why do you freeze? Because the amygdala has been overwhelmed with emotions, stressful emotions, and it hijacks the brain such that the prefrontal cortex is not able to function properly. In short, your brain is always under the control of the prefrontal cortex. We've said that is where you, the person, you are housed there. Your brain is always under the control of the prefrontal cortex. That's why you're able to think. That's why you're able to rationalize everything you do. But once the amygdala, which is mostly involved with emotions, takes over, you're not able to think properly. So you freeze because the amygdala has hijacked the brain. Yeah. And to elevate this hijacking, that the breathing in, breathing out, how we tell people breathe in, breathe out, breathe in, breathe out, that relieves the amygdala hijacking. So there's actually a scientific component to breathing in and breathing out. When you're stressed, when you're anxious, you're about to make a presentation in front of the class, and then your heart is beating so fast. You breathe in, you breathe out, and your brain, your prefrontal cortex takes back control of your brain. Yeah. So hippocampus, memory, amygdala, mostly to do with the responses, stress, anxiety, emotions. Emotions are here. Then now to the limbic system. To the limbic system. What are the components of the limbic system? And don't crack that joke of upper limb and lower limb. What are the components of the limbic system?
Hippocampal formation, and then there's the limbic cortex, the amygdala complex, and the nucleus accumbens.
Yeah. So those are the components of the limbic system. And now, the limbic system, it has three functions majorly. Number one, it mediates the sense of smell. Number two, plays a role in memory formation. We've seen the hippocampus is also forms part of the limbic system. And number three, we've seen the amygdala also forms part of the limbic system. So emotional behavior, any emotions, fear, motivation, whatever it is, right? So those are the three major functions of the limbic system: smell, memory, emotions. Okay, smell, memory, emotions. Then now let's go to a very key organ of the brain, or a very key part of the brain, that is the thalamus. The thalamus, this is where all sensory information, except for the olfaction, this smell function, but all other sensory information, this is where they come. This is where they dock, right? So whatever it is you're seeing, whatever it is you're hearing, anything, whatever it is that is touching you, all that information goes to the thalamus. Then the thalamus is what will decode and decide which region, what impulse will reach the brain. We always say that you always see your nose. Your nose is in your visual field, but you never see it, right? That your eyes are always seeing your nose, but you don't see your nose because the brain has chosen to ignore that. The other thing is that when you wear clothes stimulate your sensory receptors, right? But you're not always feeling clothes on you. You're not always feeling the clothes that you're wearing, because there's that the thalamus deciphers all this information and sends what is relevant to the brain for processing. For processing. So the thalamus, or rather, not even the thalamus, even when you're walking through town, an example is you're seeing a lot of things, you're hearing a lot of things, right? A lot of things are happening. But if someone somewhere mentions your name, you'll be able to hear them, despite there being a million activities happening, right? When you're sleeping in class and the lecturer mentions your name, you will wake up. Why? Because the thalamus has deemed that this particular information, this is relevant. This is key. This information has to reach the cerebral cortex. Okay. So the thalamus is where all that sensory stimulus will dock, and the thalamus will now be the one to convey the information to the higher centers of the brain. Okay. So it will play a critical role in arousal, in learning, in memory. The thalamus literally is like a mini brain. It has a lot of roles. You see, even awareness, even emotions related to the sensory experience will be they'll be decoded here in the thalamus. The hypothalamus, I believe the hypothalamus, we mention it a lot. We know about the hypothalamus. We know it will stimulate the various hormones of the pituitary gland, and these hormones of the pituitary gland, water, sexual responses, endocrine system functioning. It has a role in the sleep, when it is sleeping and waking up. Body temperature regulation, we know the gun, we call it the thermal gun. You point it to the thalamus so that you can know what temperature is the thalamus setting for the rest of the body. Same applies, or rather, that's the paracetamol that we use for fever. We know it has to do, what it inhibits is this prostaglandins elevating the temperature at the hypothalamus, because the prostaglandins, they'll tell the hypothalamus raise the temp, but your paracetamol there, that is what it will inhibit. So the hypothalamus is another key area. The brainstem, this one we discussed it in detail in the other discussion about the midbrain, the pons, the medulla. We say the autonomic nervous system, especially, we'll see it being housed here at the medulla oblongata. Things such as breathing, things such as the heart rate, it will all be controlled here. Yeah, cardiac center, respiration center, vasomotor center, the vomiting center, hiccuping, swallowing, coughing, all of these things, yeah, they'll be housed here. And then the pineal gland, what hormone does the pineal gland produce, anyone?
It's melatonin.
Yeah, melatonin. Melatonin, you know, melatonin plays a role in sleep. Yeah, sleeping and waking up, right? And what do we say controls that pineal gland? There's a rhythm. What rhythm controls that pineal gland?
Circadian rhythm.
Yeah, circadian rhythm. Yeah. Essentially, yeah, the circadian rhythm, your brain has sort of a clock, right? And the clock, based when it gets to nighttime, more melatonin is released. When it's approaching in the morning, less. Yeah, we know about the circadian rhythm. It's simply your brain clocks, or your clock's brain. I don't know. Yeah. Then the spinal cord, when we're talking about the spinal cord, here we mentioned two matters, the gray matter and the white matter. Yes, this one, I think we've mentioned it before, whereby even a picture was sent to the group with all of this, the differences. The gray matter is the cell bodies and the dendrites, and the white matter is more of the myelinated axons that will carry information from the gray matter to the brain or other areas of the spinal cord. Dorsal root ganglion, you can see. Now, this is the sensory nerve here. It has brought the information. The information is deciphered, and then the motor nerve takes the information to the muscle, telling it, maybe do this, don't do this. Here, you'll see the reflexes, the brain reflexes that are there. Yeah. Then something to mention about when we are mentioning the varicella zoster virus, we mentioned that the varicella zoster virus, it establishes latency. Latency in that it will go to your nerve and relax there and wait for the day that your immunity is down and attack, right? So the varicella zoster, usually it's here in the dorsal root ganglion. It will go and relax there. Then one day it will be activated, and then we'll see shingles, right? Yeah. Then we go to the meninges of the brain. We know these ones, they're there to offer protection. And we say DAP, or DAP from outside inwards. We have the dura mater, the arachnoid where the CSF will flow, and the pia mater. The dura mater, this is the thickest layer of your meninges. The dura mater, it's the thickest layer of your meninges. So number one, of course, being the thickest is the one, protection is a major thing. The venous channels for brain tissue blood drainage will be found here in the dural sinuses. The arachnoid, we already say this is where the CSF is flowing. And then the pia mater is what carries blood supply of the brain. In the pia mater is where we'll see our capillaries from the circulatory system. Yeah, we already talked about the ventricles, but the CSF will flow here in the ventricles, right? Seraphine already mentioned that synthesis is by the choroid plexus through filtration of the blood, right? And yeah, once produced, the CSF will move freely through the ventricles. I think we've already mentioned all of this. Yeah, you said it's been filtered. You filter the blood, and you obtain your CSF. Yeah, comes by filtration of the The 12 cranial nerves. I think we discussed the 12 cranial nerves before, and unless someone wants us to go through the 12 cranial nerves, but there's that mnemonic that was sent, or even that we were taught by our anatomy lecturer, to touch and feel very good, VAH. So I hope you remember that mnemonic. To touch. Feel.
Guys,
Yeah.
someone said kindly go back to the cranial. She wants to take a screenshot.
To touch and feel very good, VAH, olfactory, optic, oculomotor, the trochlear, the trigeminal, abducens, facial, this vestibulocochlear. I think this is an old way statue acoustic, or vestibulocochlear, glossopharyngeal, vagus, accessory, hypoglossal. Now, if we are just to look at the names, maybe olfactory, smell, optic, vision, oculomotor, it will control eye movements and pupil constrictions. Then abducens, I'm jumping to abducens. This one will control also eye movements. Same with trochlear, trigeminal. We know the trigeminal divides into 3 major branches. Which are the 3 major branches of the trigeminal nerves?
Ophthalmic, maxillary, and mandibular.
Yeah. The ophthalmic, the maxillary, and the mandibular, right? And even now from the names, you can be able to decipher their function. Right? Ophthalmic will relate to the eye. The mandibular is where the jaws will be. The maxillary, what does the maxillary do? Which organ is it involved with? Anyone? Maxillary? Assignment for the day.
Is it for pain? I'm not sure. I'm just reading.
It's sensory, yeah, but to what area? What area does it supply?
Jaw. Apa.
Pharyngeal.
Me, I don't know. I'm asking. I'm also asking.
I think it's like the cheek area.
The
And then
cheek.
and then the mandibular is now for the jaw.
For the jaw, yeah.
Yeah.
Nice. So before we go to the pharmacology of neurotransmitters, there are some critical parts that we need to look at. We haven't mentioned the structure of a nerve and how an action potential is transmitted, all of those things. But before we go into that, we say that there are two types of cells, generally two types of cells in the brain, the neuronal and non-neuronal, or rather the neuronals, the neurons, and the helping cells. Who can remember the types of helping cells, also known as glial cells? The types of glial cells. Alimia's saying that this is the astrocytes.
Then the oligodendrocytes, something like that.
Yeah, the oligodendrocytes. Yeah.
Pericytes.
Pardon?
Pericytes.
I'm not hearing well. I'm just hearing the sites. Microglial cells. Oh, yeah, microglial. Oh, sorry. Yeah. So I think we've mentioned the four major types of glial cells. Remember this, we call the glial cells, these are the supporting cells, and they form the majority of the cells in the CNS, meaning the supporting cells, they are more than the neurons themselves, right? They are more than the neurons themselves. So the four major types as listed here, we have the astrocytes, the oligodendrocytes, the microglia, and the ependymal cells. So starting with the astrocytes, because now astrocytes are these are the major ones. These are the major glial cells inside the CNS. Number one is that they will form the blood-brain barrier. You know, the blood-brain barrier is key. Number two is that they'll be involved in transfer of nutrients from the blood to the neurons. If you can remember from biochemistry of the brain, remember what we talked about in the biochemistry of the brain, there was that flow of metabolic products between the astrocytes and the neuron. Yeah. So transfer of nutrients from the blood to the neurons. The other thing, already said, the blood-brain barrier. It also holds the neurons together in the proper spatial arrangement. It will also serve as a scaffold to guide neurons to their proper final destination during fetal brain development. The BBB is a major one. Also, it will be involved in the production of some neurotransmitters, especially the glutamate. Again, that one, I'll just tell you, go and revise the biochemistry of the brain. Yeah. Then the other type of the glial cells, we have the oligodendrocytes. For the oligodendrocytes, we know this one is the one that will form the myelin sheath around the axons in the CNS, around the axons in the CNS. I can see someone mention the Schwann cells. The Schwann cells, they are now the oligodendrocytes of the PNS. So oligodendrocytes in the CNS, they form the myelin sheath around the axons. The Schwann cells in the peripheral nervous system, they will form the myelin sheath around the axons. So they do the same job, only that oligo is in the CNS, Schwann cells is in the PNS, the peripheral nervous system. And then also we have the microglia. The microglia, these are the immune cells of the CNS. They are sort of like the macrophages of the CNS. They are phagocytic in nature. Yeah. So the microglia, this one is more of the defense mechanism, the defense cells of the CNS, right? And of course, because they are the immune cells, they are the only CNS cell type that can be infected by HIV. Yeah, we know HIV will infect the immune cells. Neurons, they can't be infected by HIV, but these microglial cells, we'll see that they can be infected by HIV. However, infection of the microglia leading to their dysfunction is what can lead to stuff like dementia, right? To lead to stuff like dementia, because we know immune cells have these destructive chemicals within themselves. So when they are damaged, when they release these destructive chemicals, they will lead to neuronal damage, right? This neuronal damage, is what we see in stuff, especially like Alzheimer's disease. That's why in Alzheimer's disease, we do mention that HIV can be a cause or can be a contributing factor to Alzheimer's. Finally, the fourth major type of glial cell is the ependymal cells. The ependymal cells, simply what they are involved with is the formation of the cerebrospinal fluid. This one, when you ask which one is involved with CSF formation, we just say the ependymal cells. Yeah. So moving on, and I want us now to just look at the structure of the neuron and also check out the events during an action potential. This is what we were even mentioning, the shingles rash, right? Because the stuff, I don't know if there's for example, let's say that varicella zoster virus went and chilled here. So when it's activated, it will travel along the area that this nerve supplies. That's why shingles will only involve one half of the body. So if the varicella zoster virus went and then later they become immunocompromised, we'll now see this half is this area, not even just this area, this one particular dermatome is the one that will be affected. Yeah. Yeah, so simple here, let's just look at the structure of the neuron real quick, the structure of the neuron. The neuron is composed of, here we have the dendrites, and we know from the dendrites, this is where your stimulus. This is where the information will be coming from, the signals. For example, if this is a touch receptor, when someone is touched, the information, or rather the signal, is first taken up by the dendrites, right? When it's taken up by the dendrites, the dendrites will now transmit this info all the way down now here to the axons. From the axons, they go all the way to the axon terminal. The axon terminals will connect with other neurons through now, here is where we'll have our synaptic clefts and things like that. So the structure of a neuron, nucleus and the cell body. The nucleus here it is, and the cell body here. And then the axon. In the axon, there is no stuff for synthesizing neurotransmitters or anything like that. Neurotransmitters will be synthesized here in the cell body. The axon is just, think of the axon as just a tube that relays information from the cell body to the axon terminal. This is just a tube. It's a communication tube. You can see an image there. You can see the dendrites there, and then the cell body, and then the axons are there, right? Then now looking at an action potential. Who remembers the events in an action potential?
I can try.
Mm-hmm.
That was first the synthesis of acetylcholine,
Mm-hmm.
and then the transportation and the storage, then the release of it. And then it binds to the receptor, and then the degradation of it and the reuptake. That's the final. It's either degraded or it's taken back to the transported back to the vesicles by the reuptake mechanism.
Nice. So she's talked about the events of a neurotransmitter, how a neurotransmitter will be synthesized, released, work, and be returned back to the degraded or returned back to the neuron. So what about an action potential now?
Guys, did you talk about the retrograde and the orthograde?
No, I didn't talk about it.
Yeah, so in the neurons we have retrograde transport, where proteins like dynein they take like vesicles or organelles from the terminals to the cell bodies. Like anything that has been destroyed at the terminals, they're taken back to the cell body. That's retrograde transport, and it's taken by dynein proteins. And then there's orthograde transport. Now that's from the cell body to the terminal. So that is with kinesin proteins, which now they take like vesicles or proteins from the cell body to the terminals in the neurons.
Yeah, nice. Thank you for the addition, Hiral. Yeah. Anyone else with something to add now on action potential? How does an action potential happen? Say all or none. What does that all or none mean? How does it happen?
I can try.
Sam.
Okay, so basically an action potential usually is an electrical impulse that is generated due to the movement of ions across the membrane of certain excitable tissues. So in this case, nerve tissue. So we usually have certain ions that are usually separated by the membrane. So usually have inside the cell, we usually have certain ions, and it's usually negatively charged, mostly because of the proteins, some bicarbonates, some chloride ions are usually found more inside the cell than outside the cell. Outside of the cell, we usually have positively charged ions, mostly the major one being sodium, and it contributes to the positive nature of the extracellular space. So during the generation of an action potential, we usually have ion channels, the sodium ion channels opening up and allowing sodium to enter into the cell. So we said the inside of the cell is usually negative. So from this diagram, you can see we usually have the resting membrane potential to be sometimes, yeah, for nerve cells, I think it's usually around -70. Muscle cells, you'll find them having around -90 sometimes for the skeletal ones. So when the sodium channels start to get into the cell, they basically make the cell, the inside of the cell more positive. So when the charges inside the cell reaches a certain threshold potential, then we have voltage gated sodium channels opening, and then a process called depolarization, where we have the first phase of the generation of action potential. So after that happens, we usually have, this one usually happens in milliseconds. So you have the potassium channels opening, and potassium leaks out of the cell. So when that happens, the inside of the cell starts to become more negative again. And this leads to this phase where we call repolarization. So sometimes during the repolarization phase, we have a lot of potassium coming out of the cell, and the membrane potential actually goes below what it was at resting potential, and we usually call this the hyperpolarization. So I think if you were to look at this diagram that is displayed over here, that's blue region on the right side of the downward slope. Yeah, basically that.
Yeah. That's it. That's action potential. Yeah, he's covered everything. We have the depolarizing triggering event, for example, based on the type of receptor it is. If it's, for example, a thermoreceptor, the temperatures are getting high or low, it will trigger it, and then it will reach a threshold potential. In the threshold potential, you have your sodium channels being opened, and it will go all the way to plus 30 millivolts, or now here is where it will plateau. So at this 30 millivolts, or at the plateau, the sodium channels will close, the potassium channels will open, potassium will flow out. Then we always say potassium channels, they are slow channels, they take time to close. So once we've achieved the resting potential, while the potassium channels are closing slowly, some more potassium flows out, leading to this hyperpolarization. Then eventually it closes, and we have the sodium potassium ATPase that will return the potential back to the resting potential, which is usually -70. Yeah. Then now let me see, there's someone here on the chat box. Scooby-Doo, yeah. Scooby-Doo, stimulation, depolarization, plateau, repolarization, and hyperpolarization. Yeah, nice. So those are the events we've mentioned. Yeah. So who can tell me about the refractory period? We're always saying that there's a refractory period. There's some time that nerve cannot be stimulated. At what point does it occur? refractory period.
Another action potential cannot occur when during the refractory period.
Yeah, nice. Yeah. So, and there are two types, or the refractory period can be divided into two. There's an absolute refractory period and relative refractory period. So what's the difference between the two? The absolute refractory period and the relative refractory period.
The absolute one is that's the time immediately after. So during that time, there is no way like a new action potential can be generated. Like there's no way. But then for relative, as much as it's hard to generate an action potential, it's actually possible, but then you'd require a stronger stimulus than the original one.
Yeah, perfect. In the absolute, absolutely no way another action potential can be generated. You can see even from the diagram, you can be able to know this is the absolute refractory period. This is a stage whereby we have our neuron being depolarized and being repolarized. At this stage, there is no way that you can generate another action potential, because you can say the neuron is still, it's in the active phase of the rising phase. But in the relative refractory, you can see here the neuron has gone back to, it's a bit hyperpolarized, right? But as Seraphine has mentioned, we need a very strong stimulus, stronger stimulus than this one, to generate an action potential. So the absolute refractory period is whereby it is simply impossible for another action potential to be generated. But in the relative refractory period, it is possible. However, the triggering event that we mentioned when we were generating an action potential in the beginning has to be considerably more potent than usual, Yeah. So if, for example, if you've been slapped, for you to, if the person slapping you is fast enough, for you to feel the second slap, they have to slap you harder than they slapped you the first time. Okay. Yeah. Unless someone has something to add on neurons, I'll request now we go to the neurotransmitters, unless someone has a burning something to add. I know there's a lot that we haven't covered, but we've covered the basics. Myelin sheath, and what's the effect of one neuron having a myelin sheath and one other neuron not having a myelin sheath? Which one is better? Which one is faster? Let me see which one is faster. Between a myelinated and a, let's say, a demyelinated or an unmyelinated.
Myelinated would be stronger. Faster.
Yeah, myelinated would be faster. Why?
Because the myelin sheath is an inductor, so the impulses would just pass right through them.
Yeah. So here it's just the impulse is from one node of Ranvier to another. It's just jumping and jumping and jumping. That is where another action potential is happening. But in a non-myelinated, let's just go back to this. There's an image here. Yeah, this is a non-myelinated. You have to keep depolarizing, 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 depolarizing all the way to the axon terminal. But for a myelinated one, you just have to this yeah, you just have to, there's this area that you don't need to depolarize, a node of Ranvier, and then this area, and then this area. So of course, this one will be faster because we don't have to keep depolarizing this here, because fat is an insulator. The current will travel all the way until here, then we'll depolarize here, kiasi, travel, see? So this one will be faster. This one will, the current here will travel very fast, yeah, as compared to a non-myelinated, or yeah, non-myelinated.
Guys,
Yeah.
I have a question.
Uh-huh.
The autosomal disease, autoimmune disease, multiple sclerosis, it affects the myelin shields of the axons. So as we have said earlier, the glial cells, the oligodendrocytes, they form the myelin shield. Can we say the multiple sclerosis, they affect the formation of myelin shield from the oligodendrocytes?
Yeah. Here it is. Yeah. There's that that in multiple sclerosis, there's that loss of myelin, right? Because the immunity has attacked this producing, these cells are producing that myelin shield. Yeah, so you're good, yeah. What you're saying is correct. Anything else maybe someone wants to add, or before we go to neurotransmitters? Because now we are just going to neurotransmitters.
EMS tulisema ni class 2.
MS. I think ilikuwa class 2, siko sure. Maybe someone can confirm. What
I think
type
it
of
is.
sensitivity reaction it is? Siko sure.
I think we also said it's class 2.
It's class 2.
Yeah, I think.
I'm seeing as a type 4 because the Nini is mediated by the T cell.
Hello.
Hello.
Yes, I have a question.
Mm-hmm.
So how do we differentiate the type 2 and type 3? Because especially the autoimmune conditions, do they fall under type 3 or it's type 2? Because there are some which we classified under type 2 and some under type 3. So when do we really know when an autoimmune condition is supposed to be where exactly?
I think now for that, it will be based on the pathophysiology of that particular disease. Because in type 2, we say that is antibodies are attacking surface antigens. But type 3 is more of deposition of immune complexes to certain areas such as joints, such as the kidney. So we said Graves' disease will be a type 2 because there is that antibody against the TSH receptor or something. And then rheumatoid arthritis will be type 3 because some immune complexes can be deposited into the joints. So my thinking, my thoughts, it's you have to focus on the exact pathophysiology of the disease. But those are my thoughts. I don't know if Stanley is here, is the one who discussed the hypersensitivity reactions. Or anyone who's an expert on hypersensitivity reactions.
Okay, so I think MS is type 4 because it's the T cells that attack the myelin sheath.
Yeah. So it's type 4. Not that myelin. Yeah, because it's a cell-mediated hypersensitivity. Yeah. So quickly, we'll just go through now the neurotransmitters. For neurotransmitters, Shafi already gave us a summary about how neurotransmitters happen. They are synthesized, they are stored, they are released, they bind to the receptors, and then they are inactivated or they are recycled, right? And a good example was acetylcholine. It will be synthesized, stored, released, then act on the nicotinic or muscarinic receptors. Then it can be inactivated by the acetylcholinesterase, or it can be reuptake, be taken back, be recycled. Yeah. So neurotransmitters, they can be classified based on their chemistry. We have the biogenic amines, that is the serotonin and the acetylcholine, and then the catecholamines, dopamine, norepinephrine, and epinephrine. So the biogenic amines, acetylcholine, and serotonin, then the catecholamines. I believe the catecholamines, we're all familiar with them. Dopamine, norepinephrine, epinephrine. Yeah. Then also we have amino acids as neurotransmitters, such as GABA, glutamate, glycine, aspartate. Yeah. GABA and glycine, what's common about them? And what's common about glutamate and aspartate?
I think GABA and glutamate are inhibitory.
GABA and
Glutamate.
now glutamate is excitatory. Glycine is inhibitory.
Okay.
Then yeah, so GABA, glutamate and aspartate excitatory. Then also the neuroactive peptides such as bradykinin, substance P, vasopressin, oxytocin. Yeah. Then the core transmitters, which these are stuff that will be released with the neurotransmitters, such as the VIP, vasoactive intestinal peptide, ATP, adenosine, all of this stuff. So we'll study neurotransmitters using acetylcholine. So first of all, we said we have to synthesize. To synthesize, we'll need the various precursors. So for acetylcholine, we need choline. The acetyl component will come from the acetyl-CoA. Yeah. And then choline will be formed from serine. You see, serine converted to choline using that serine decarboxylase, then all of that. So acetyl-CoA plus choline, now we have our choline acetyltransferase, and voila, we have our acetylcholine. Drugs already here, we're talking about drugs. For example, a drug such as hemicolinium, what it can hinder is the transport of choline from the plasma into the neurons. So here it is. Remember, choline, here it is. In the neuron, we are having acetyl-CoA and choline being acted on by this enzyme, choline acetyltransferase. And this choline is coming from the blood. So one way to inhibit, or rather, if you are looking to inhibit the action of acetylcholine, you can block this choline transporter here using this drug hemicolinium. There won't be choline available for synthesis of acetylcholine, and you will have reduced the action of acetylcholine. The other place of inhibition can be on this this transporter here that transports the acetylcholine from the cytosol, quote unquote, into the vesicle. Vesicular amine transporter, vesicle associated transporter, yeah. So if you block that, acetylcholine will just be hanging around. It won't be inside a vesicle to be released in case of an action potential. Yeah. So transport and storage, we already looked at that transporter. It transports it from the cytosol into the storage vesicle. So there's a drug that can inhibit that. Then we know the release of a neurotransmitter. Here we're talking about there's been an action potential. Calcium has gotten in. When calcium gets in, we know it will allow the synaptic vesicles to move to the presynaptic membrane and release their contents, right? So the release of a neurotransmitter, I think that is common to all neurotransmitters. An action potential will activate the voltage-gated calcium channels. Calcium gets in and stimulates the fusion between the vesicle and the plasma membrane, essentially releasing its stuff. And there are proteins that will be involved in that docking into the plasma membrane and fusion, such as the snare protein, the synaptobrevin. Now, these proteins are significant, especially when we're talking about the Clostridium botulinum. Is it botulinum? Yeah. Yeah, we'll see them there. Oh, they are significant then. Release, here it is even. So we know Clostridium botulinum and Clostridium tetani. They are both in the Clostridium family, but one causes flaccid paralysis, and the other causes spastic paralysis. Which one causes what? Based on even what has been described as the mechanism of action here. One causes flaccid, the one
Spastic paralysis is tetanus, and the other one is botulinum toxin.
Yeah, very nice. Now for flaccid paralysis, it means we don't have enough acetylcholine. For spastic, it means we have too much. And that is how they work. Clostridium botulinum, it will hydrolyze this snare complex. Where have we seen the snare? Let me just take you back here. Here, it is simply, just remember this snare proteins or complex, they'll be involved in the release of a neurotransmitter. So once the botulinum hydrolyzes or inactivates them, there won't be release of the neurotransmitter. In this case, we won't release our acetylcholine. So leading to that flaccid paralysis. However, for tetany, we transmitter. So what Clostridium tetani does, it inhibits the inhibitor, right? Remember that analogy. Once you inhibit the inhibitor, you lead to more activation, right? Because glycine, what the glycine interneurons were supposed to do, they were supposed to be the balance of the contraction. Acetylcholine has gone and activated a contraction, and then the glycine is supposed to check and balance. But once you inhibit the glycine, it means acetylcholine action goes unchecked, right? There's no balance for acetylcholine. There's no check for acetylcholine. Hence, there's this prolonged contraction, right? Or that spastic paralysis, sometimes also known as the locked jaw. Another example of a toxin that acts on this nervous level based on the neurotransmitter is this tetrodotoxin, which is released by this fish, some types of fish, which blocks the sodium channels. If we block the sodium channels, there won't be any action potential. If there's no action potential, it means we won't be releasing any neurotransmitter. Okay. So we already said this, tetanus. Then now binding here. Now on binding, this is where pharmacology gets in, right? Because now we are talking about receptors. We've released our acetylcholine, can bind to a myriad of receptors. There's the nicotinic receptors, N1 in the neuromuscular junctions, the one we use for contraction. We have the nicotinic receptors in N2, the ones in the brain and in the adrenal medulla, right? Then muscarinic receptors, yeah, M1 all the way to M5. We know the muscarinic receptors, these are the ones that will be found, the M2, the ones that will be found in the heart, right? And will work to stimulate or, yeah, to inhibit those. Finally, we have the degradation or breakdown of acetylcholine. Now here we have the enzymes like the acetylcholinesterase, which will break down acetylcholine, essentially leading to an end of its function. Whatever function it was doing, once we break it down, it can't function, it can't stimulate any contraction any further. And at this point, well, I'll ask the question, how do organophosphate poisons work? What is the mechanism of action of organophosphate?
They usually covalently bind to acetylcholinesterase using the phosphate molecule of the phosphate group. And when they bind covalently, then it means that you don't have something to degrade the acetylcholine. So acetylcholine will be excessively expressed in the receptors, leading to the muscarinic effects we see in the poisoning, such as excessive excretions, pupil constriction, and all the others.
Yeah, perfect, yeah. They irreversibly inhibit this acetylcholinesterase. So acetylcholine action is prolonged, leading to those symptoms of OPP poisoning that she's described: excessive secretions, pupil constriction. Someone can have the diarrhea because of increased crying, sweating, excessive sweating, or not crying. We were told it's lacrimation, not crying. Crying is an enemy. And what's the antidote of organophosphate poisoning, and how does it act? Now we've seen the mechanism of action of the poison. What's the mechanism of action of the antidote?
Okay, so there are two antidotes that can be given. So atropine, just the basic one. This one usually acts by inhibiting the muscarinic receptors, basically the acetylcholine receptors. So by inhibiting them, then you prevent the muscarinic effects. Then the other one is pralidoxim or other oxins that can be given. This one's they help to remove the organophosphate from acetylcholinesterase, but then they should be given before the process of aging occurs. And this is basically whereby the complex usually loses methyl group, and when it does that, it becomes tightly bound and cannot. That means if you give pralidoxin, the antidote won't work. So it's usually supposed to be given before aging occurs. But it basically helps to remove the organophosphate from the acetylcholinesterase enzyme.
Yeah, perfect, yeah.
Then
There
are two types of organophosphate. There are reversible enzyme, like zeostigmine, neostigmine, and then the reversible will be parathione, and then the nerve gases like serine, those are irreversible. And then the reversible one, it's the one that mostly we use the palidoxin, since it displaces the organophosphate from the receptor, and then it will bind to the receptor and reduce the effects of the organophosphates.
Yeah, nice. So yeah, she's brought the two types of acetylcholinesterase inhibitors, the reversible ones, which are usually our drugs, and the irreversible ones, which are usually the poisons. And I think yesterday or the day before that, we said pyridostigmine, neostigmine, they can be used to treat a certain autoimmune condition. Which condition was this? Yeah, nice. Yeah, myasthenia gravis. Yeah, nice. Yeah, breakdowns we've already mentioned. All of this is mentioning the reversible and irreversible inhibitors. Yeah, myasthenia gravis, it is. So we've seen this. And then the other thing that can happen is the reuptake, right? Once acetylcholine has been released, binded, it can be taken back into the various back into the neurons. There are those uptake, and especially for the uptake, just going to skip all this. We especially see them with things like the serotonin. That's why you have in any SSRIs, right? And even norepinephrine. Because for acetylcholine is not that major, but for these catecholamines and that, yeah, exactly. There's this norepinephrine transporter. And if you inhibit this transporter, there'll be more norepinephrine at the synaptic cleft. And we know this has been utilized for the treatment of depression. Drugs such as the tricyclic antidepressants, they inhibit that norepinephrine transporter, hence more norepinephrine at the junction, at the cleft. So, and we know about the theories of depression, that there is a reduced norepinephrine, there is the reduced serotonin, hence the mechanism of action. Yeah, another thing you can see, cocaine, it also inhibits these transporters. So cocaine leads to that more norepinephrine on the synaptic cleft. Hence, cocaine can be used as an antidepressant. True or false? Okay. The other part is just the receptors of the various neurotransmitters. For that part, I'll just leave you to go through that by yourself. Essentially, that is it from me for neurophysiology, unless someone has something to add or would like us to go through something specifically. That is it. If no additions, no questions, then we'll stop there.
Guys,